Alrighty, good morning, and we are going to do chapter 11 today, human genetics. This lab is mostly a paper lab in that it is pretty much all problems. So there really isn't anything we would have done in the lab. So this is more of just homework. So the key things that we're going to talk about here is non-disjunction. And so when we talk about non-disjunction, what we're talking about is the problem where meiosis or meiosis failure, where things do not go right. And there are two main places that things can go wrong. The first is in meiosis 1, and that is where the homologous chromosomes do not separate. So we fail to have what I call the divorce. Mom and dad did not separate. The second place would be in meiosis 2, and that would again be where the sisters don't separate. So both of these would mostly be like an anaphase 1 and anaphase 2, or they don't move apart. All right, so, and I kind of show you this in the next slide. So here we have non-disjunction at meiosis 1. The homologous chromosomes come together, and now our gametes have one extra chromosome here, one extra chromosome here, one less. So this would be n plus 1, n plus 1, n minus 1, n minus 1. So it would be shorter chromosome. Over here we have disjunction, or I guess you can call it disjunction, but I would say segregation. Remember we talked about the laws of segregation with Mendel. So we segregated our homologous chromosomes. The divorce was successful. However, here we don't have the sisters did not separate. So the sisters separated fine in this one. So we have the correct number of gametes, 23 and 23. But this is where we had failure to separate. So we have non-disjunction with the sisters. So we have n plus 1 and n minus 1. And that is what we are looking at the first part of this lab. We're looking at separation, and specifically we're keying in on the sex chromosomes. So what we're saying here is that we have a male, right, X and Y. We're going to make sperm. And so the first thing we have to do is, remember we're in cell cycle, we have to finish through interphase. So we go through synthesis and make the sisters. So we're writing this as XXYY. So, and these would be attached, and I kind of have that in some of the videos that I put in there for you. So I'm writing it, if there was a female, there'd be 4 X's. And again, the 2 X's, they'll be hooked together because, right, that's a duplicated chromosome. So we have our separation of our homologous chromosomes, and now we have to separate our sisters. And this would be a nice, healthy production of gametes, in this case, spermatogenesis. So we made our 4 sperm with our correct chromosome number. Now, in your lab book, they have you going ahead, and it asks you, the first thing it asks you to do on page 152, on page 152, it asks you to have non-disjunction in meiosis 1. So they all went together and they did that for you. So notice that we're going to be n plus 1, n plus 1, n minus 1, n minus 1 here. And then they show you again here, and there are so many different ways we can do this. So here we have proper segregation at M1, but here we have issues. The sisters in this case did not go together. Now we could put them down there. We could have had the X's not undergo separation. So there's all kinds of ways that we could draw it. So there's this or. So we could have this, we could have that, we could have, so there's all these different possible combinations that we could have. So here we didn't separate both of them. So n plus 1, n minus 1, n plus 1, n minus 1. Or we could have where these separated okay, so that would be the correct number, n minus 1, n plus 1. Or we could have the correct number, n plus 1, n minus 1. So there's multiple ways that we could have done this. All right. So then on the next page, on page 153, they ask you to do this with the female chromosomes. All right. So again, there are multiple ways that we can do this as long as you understand what we're getting at here. So what I have here is at the top one, we separated correctly. So our meiosis 1, and that's what it asks you. It asks you for non-disjunction of meiosis 2. So we separated correctly in both cases. In this case, I did not separate the blue, the red, the red X's. So again, one X is for mom, one X is for dad. So pink, blue, try to color associate that. And then in this case, I show you, well, dad's X chromosome did not separate correctly. So again, you can pause this and then copy this down. It's also in the book review too, which is not a video. So then it asks you, based on that picture, what are the possible gametes? And again, so that's where you're going to say, well, you're going to have N+1 or N+2 with meiosis 2. Right. Now you could just say N, because the other one, the other chromosome that separated may have been normal, but in the case of the side that we did not have our sister separate, we'll have one extra or one not enough. And so then it says, well, what would be the result of combining the gametes with the above question with a sperm carrying an X chromosome? So if we had a sperm carrying an X chromosome, right? So we would have our N, so we would have N+1, so we'd have XX, right? This would be from mom. So mom would have the 2 X's because she had the 2 extra ones. And then this would be dad's contribution. So this would be, we would have a super female, XXX. Now, that would be this case. Now, if we did this one, we'd have O, meaning it's missing, but we'd have dad's X here. So this would be Turner syndrome. So we have an XO child. And then it asks, well, what if we had a Y sperm? So we'd have our XX from the mom if it was N+1, and then we would have the Y from dad. And so that would be Klinefelter's. And then again, we'd have the O from mom if it was N-1, and the Y, we have U, all right? So, and in fact, this baby would be miscarried because there are critical genes on the X chromosome you have to be alive. So that would be bad, okay? And then it asks in which parent during the mitotic division must non-disjunction have occurred to produce an XYY, a Jacobs syndrome. So again, that X would be from mom, okay, in this case. And we got the 2 Y's, okay? So we could have the X and Y from dad if we had non-disjunction in meiosis 1, but the problem we would have is, well, where did the other Y chromosome come from, right? We know it can only come from dad. So where did the extra Y come from? It had to come from dad. And it had to come from, because the X had to come from mom. So it would come from non-disjunction in meiosis 2, where the sisters did not separate, okay? And that's what you would be filling in for the end at the page 154. So again, you can pause this, write this down, take your notes. I'm going to move on. The next thing that your book talks about are different, did I not do that one out? Or is that a repeat? Oh, I think I went the wrong way. There we go. All right. So, the next thing your book talks about is defective alleles. They go in and talk about 3 different cases. They're going to talk about, so if you're with me on page 155, they're going to talk about autosomal dominant, autosomal recessive, and X-linked recessive. So what we're saying here is autosomal means that you have 2 chromosomes, you have 2 copies of those genes. Autosomal dominant means you only need one bad copy and you have it. And we kind of did this with our Huntington's disease in our pedigrees. And another one would have been, I talked a little bit about achondroplastic dwarfism is also dominant. And polydactyl, believe it or not, 1 in every 500 people are born with an extra finger or a toe. Generally, that is a very small, kind of like a little flap on the outside of your pinky, and they just kind of cut it off at birth. So you may not even have realized it, but it is a dominant disorder. So we also have what is called, easy guys, we have autosomal recessive, and autosomal recessive is where you need 2 bad genes in order to have it. Okay, so that means, again, so remember, autosomal dominant means you're not really going to have a carrier, because a carrier means you have that bad thing or that gene that we're talking about, but you don't express it, you don't show it. With a dominant disorder, you show it. With one copy, you show it. So you can't really have what's called a traditional carrier. Now, in autosomal recessive, you could be a carrier, meaning you and your significant other could both be hiding this bad gene. And when you have a child, you have a 25% chance then of having a child that would get each of a bad copy from each parent and then have that disorder. And so we talk about things like Tay-Sachs, and we talked about cystic fibrosis. So those are some albinism. There was a whole list at the end of that presentation. And then what we've been working on recently is the sex-linked characteristic, which means, and she has specifically said X-linked recessive, meaning it is on the X chromosome. There are some genes on the Y, but we really don't get into it in this class. We're focusing on the X. Every human has an X. Women have 2, men have 1, which is why men cannot be carriers either. They either have it or they don't. And so we're going to look at that. Now, when you do your autosomal dominant and recessive pedigrees, you have to understand you will be using letters, 2 letters for those 2 copies. But when you are doing your sex-linked, or in this case, X-linked recessive, you're going to be using those X and Y chromosomes with superscripts. So make sure that you are doing that. And then on page 156, there are just genetic problems for you to work out. So what I would highly suggest, and I always do this with many of my classes that I take, is I would take a picture or a scan of the blank page, and then I would print out several copies. And then I would work the problems, look at the key, which I have in the module, and then correct them, and then try it again and see if you get it right again, and just keep practicing them until you do feel confident that you know how to do these genetic problems. Now, the pedigrees are a little bit more, a little harder. Now, we had a pedigree assignment in here. If you didn't do it, I would highly suggest you doing it, checking your work with the key, figuring out what you're doing right, what you're not doing right. You will be asked on your lab practical, you will be given pedigrees, and you need to tell me what is the inheritance pattern. Now, what I mean by inheritance pattern, is it autosomal dominant, is it autosomal recessive, or is it sex-linked? So you need to be able to identify this. Now, things that I look for when I look at a pedigree, I look at, for example, right here, both of these parents are normal, and yet they have 2 children that have it. Now, again, this cannot be a dominant disorder, because if this individual has it, one of the parents also have to have it. So I've just ruled out autosomal dominant. Notice I have equal numbers of boys and girls that are displaying this disorder, therefore it is not sex-linked. So it is possible that both 1 and 2 are carriers, and that is how they have 2 children with it. We can look here now, of course, since this person has it, 10 will be getting 2 bad copies, and, or sorry, 1 bad copy, right? 1 bad copy from mom. And so we know that this guy here is a carrier. His wife has it, which is why they have 2 out of their 3 children have it. So this is in fact a autosomal recessive disorder. So you are expected to know that. You will then need to fill in the genotypes. So I would go in and practice doing that. Now, since we know it is an autosomal recessive, or we can then start filling things in. So like for number 7, we tend to use A's. So that would be little a, little a. Like I said, over here, we have this guy here has to be big A because he's normal, and a little a because his mom has 2 little a's, only has 2 little a's. So I could continue filling this in. You need to know how to do those types of things. You need to understand the genotypes and the phenotypes and to fill those in. Now the next pedigree, when I look at the next pedigree, I can get my page to move forward. Now look at this one. Look at all the boys. It's only the boys that have it right there. That should jump right out at you that this is sex linked. And again, when I fill this in, I have to use my, so up here, I have to use, well, use XY, but then I'm going to have to use that superscript, and I don't know how to do that on this program, but so I'm just going to put it here in a parenthesis. So he would be little a, so again, that would be superscript A, Y, all right? So he has it, right? So he's got the recessive disorder, which in this case could be color blindness. All right, so we know that, another example here, which one do I want to do? Oh, let's do, well, dad, let's do dad. This dad, number 5, he has to be big A, Y, right? He's normal. So where did those 2 boys get that bad copy? Well, we know that dad gives the Y's to his boys. So this mom right here, she is one of those sneaky, sneaky carriers I talk about. So she would be X superscript A, X superscript little a. So she is a carrier. Okay, so, and again, I would practice writing this out. I would, if you don't want to make multiple copies or whatever, or just do it in pencil, so you can go in and fix the ones that are wrong. So I would fill it out, check with the answer key, and then try to figure out why you got that wrong. And then you have, I did 2 of the 3 pedigrees, so you should be able to figure out the next one. I didn't want to give it all away, so you have some practice. And again, like I said, if you didn't do the pedigree homework or practice, you may want to go back and do that, because you need to be pretty, I don't want to say fluent, but you need to be comfortable with this, because you don't want to spend 10-15 minutes on a problem. You'll run out of time on your practical. All right, so that is it for this lab. So now all you need to do is, again, I would highly suggest watching the videos. I would highly suggest checking your answers and then doing the review, because once you are at that point, you should know what you understood and what you did understand, and then go ahead and do the review and submit that. Hopefully this is helpful, and I will be making the next and last lab video here soon, probably later today. All right, thanks, bye.